Okay, very good evening, my dear friends. Today's our eminent resource person, Dr.A.SHYLAJA Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous SIDDIPET. Madam, please welcome and start your session.
Okay, sir. Thank you. Very good evening to all. Today I am going to discuss about the ARCHEAE bacteria, ACTINOMYCETES and MYCOPLASMA. So first, ARCHEAE bacteria. ARCHEAE bacteria it coming from the two Greek words, that is archaeas and bacterian. So I will share the screen first. Okay. Sir, is it okay?
Yes, madam.
Visible?
Yes. Yes, madam.
Okay. So the ARCHEAE bacteria, it coming from the two Greek words, that is archaeas. Archaeas means ancient, and bacterian, it's a small rod. Bacterian means small rod-shaped structure. These are the heterogeneous group of bacteria with prokaryotic cell structure, but diverse in their morphology and physiology. So same, it's look like a bacteria, but they usually prefer restricted or extreme aquatic or terrestrial habitats. They often occur in anaerobic and hypersaline or high temperature environments. These ARCHEAE bacteria, generally prokaryotic organisms. These organisms first recognized by Carl Woese and G. Fox in the year 1977. Okay, ARCHEAE bacteria are prokaryotic organisms. It's a word of two Greek words, that is archaeas and bacterian. Archaeas means ancient, bacterian means a small rod. Okay, so these first recognized by Carl Woese and G. Fox in the year 1977. So first we are coming to the what are the features having these ARCHEAE bacteria. The cell walls do not contain peptidoglycan, but a range of other unique polysaccharides are present. In this cell wall, we are observed that peptidoglycans are absent and only polysaccharides are present in the cell wall. The cell membrane is a single layer of glycerohydrocarbon and glycerol chains instead of a bilayered phospholipids arranged tail to tail. Okay, in the cell membrane, we are observed that is a single layer of glycerohydrocarbon glycerol chains instead of a, generally we are observing that in the cell membrane, we are observed that bilayered phospholipids. But here in these ARCHEAE bacteria, we are observed that the layer of glycerohydrocarbon glycerol chains. Okay, the 16 microns RNA molecules are similar to each other, but differ greatly from those of other bacteria and eukaryotes. The ribosomes of these organisms are insensitive to chloramphenicol. Is an antibiotic, and the first amino acid to initiate a new polypeptide chain is methionine instead of formal methionine. Instead of formal methionine, the polypeptide chain is methionine. The first amino acid we are observed in this structure. These organisms inhabit extreme environments. They include the methane producers, methanogens are there, and the red extreme halophiles and the thermoacidophils. So based on the habitats, these are mainly grouped under three categories or four categories. Okay, the archaebacteria, these organisms grow in the extreme habitats where other organisms generally does not exist. So, based on that habitat, these are broadly grouped under 4 categories. That is halophils. Halophils means what? The archaebacteria lives in the salt water. So, where some members are lives in the salt water, that members are called halophils. Next one is the thermophils. These bacteria grow at highest temperature, high temperature. So that's why we are called thermophils. Alkaliphils. Third one is the alkaliphils. Bacteria growing on alkaline soils. Okay, that is the alkaliphils. So bacteria, archaebacteria growing on the alkaline soils. That type of archaebacteria are called alkaliphils. Next, fourth one is the acidophils. These bacteria grow in the high acidic regions. These are the 4 types based on the habitat. These are broadly divided into these 4 categories. Next one is the morphology. So we are coming to the morphology of archaebacteria. We are observed that some members are spherical in shape, some are rod shaped, some are spiral, and the plate.
Madam, slides. Madam, slides is not moving.
Only one slide
Okay.
from this.
Okay, madam. No
serious,
problem.
sir.
Please proceed. No problem.
Okay.
Madam, please proceed.
Okay. Okay. Next one is the archaebacteria. Some are spherical in shape, some are rod shaped, some are spiral and plate like shapes. But sulfolobus like Archaea species are pleomorphic or irregular in shape. So where we are observed in the sulfolobus, in that type of archaea species, there's a that are pleomorphic or irregular in shape. So the peculiar feature is the presence of lobes to the cells. Sometimes, rarely they are filamentous. Sometimes we are observed that the archaebacteria are filamentous. These cell wall made up of some members are phospholipids, some members having the already we are discussed that glycerohydrocarbon glycerol chains. And the nucleus and true cell organelles are absent but possess cell wall and flagella. These are having the cell wall and flagella. Single circular chromosome is present in the cells. See here, same look like bacterial shape. This structure, this is a prokaryotic cell. In the DNA of chromosome, only 15% genes are considered as specific, but these gene-specific action is not yet recognized in this. In Archaea, methane-producing genes and their specific actions were recognized. So that are methanogenic bacteria. We are called methanogenic. Archaea genes transcription and translation to some extent resembles the eukaryotes. That is the specialization of these archaebacteria types, methanogenic types, halophils and thermoacidophils. So methanogenic type, what are the 3 types are there in the archaebacteria? There is a methanogenic type, methanobacter. Examples of methanogenic types: methanobacter, methanococcus, and methanospirillum. See here, some are rod shaped, some are spiral shaped, some are spherical shaped. So methanobacter, methanococcus, methanospirillum, 3 are there. By reducing the carbon dioxide, methane is produced under anaerobic conditions that are called methanogenic type. Hydrogen ions are oxidized and the energy of the electrons released was utilized to produce the methane. So they are identified as obligate parasites. That is the obligate parasites, methanogenic type. Examples of this methanobacter, methanococcus, methanospirillum. Coccus are the rod-shaped bacteria and spirillum means spiral-shaped. Okay, methanobacter and methanococcus and methanospirillum. These are the methanogenic types. And next coming to the halophils. So halophils means they are growing in the salt water, high salt concentration environments. Halobacterium and halococcus are examples for this. Okay. So for the growth of these bacteria, higher salt concentration is mandatory. So that is why these bacteria are described as obligate halophilic type. Okay, first one methanogenic types are called obligate parasites, and halophils are described as the obligate halophilic type. Because of that, these are compulsory, necessary, that is mandatory. Higher salt concentration is mandatory for growing of these archaebacteria. So that's why we are called obligate halophilic type. Third one is the thermoacidophils. These bacteria grow at higher temperatures, higher concentration of acidic environments. So that's why higher temperature and higher acidic concentration, acidic environments. That's why thermoacidophils. Okay. These are the types of archaebacteria. And next we are coming to the reproduction. Reproduction occurs only through vegetative methods only. That fragmentation is very simple. That is breaking down into small bits or small fragments is the common method. And binary fission also, and multiple fission and budding. Okay, so these all are vegetative methods. That is breaking down of small bits, that is a fragmentation, and binary fission means divided into two cells, and multiple fission also, and budding. Okay, these all are the methods of reproduction of active archaebacteria. Some economic importance also there for the archaebacteria. It is used in milk industry to produce the lactose milk. It is used in milk industry to produce the lactose milk. And archaebacteria enzymes are used in molecular genetics research also. So that some archaebacteria enzymes, these enzymes are used for the molecular genetics research, and antibiotics are developed from these bacteria, which acts differently from other antibiotics. Okay, some antibiotics are also developed from these bacteria. That biogeocycles of the environment, that nitrogen, carbon, sulfur, these are carried out by these bacteria. It is a natural process by which carbon, hydrogen, sulfur elements are maintained in the equilibrium. So this is the economic importance of archaebacteria. Okay, so some methanogenic bacteria are differentiated on the basis of morphology and gram reaction. So that differently divided: methanococales and methanomicrobiales and methanobacteriaceae. Okay, so some difference are this. It's not important. And halophilic also, halophilic, thermoacidophils. These are the three types of we are observed in the archaebacteria. Next we are coming to the, see here, mycoplasma. So this mycoplasma, see here, this is the mycoplasma. Mycoplasma, it is also the smallest known aerobic prokaryote without a cell wall. Okay, without a cell wall, that is the mycoplasma. Due to lack of cell wall, the cells are pleomorphic, occurring in different shapes. So when that cell wall is absent, that is the pleomorph, so occurring in different shapes: spherical, ovoid, and filamentous. Okay, the plasticity of cells allows them to pass through the bacteriological filters, even though the smallest cells are about 0.3 micrometers in diameter. So this is the smallest aerobic prokaryote without a cell wall. So due to lack of the cell wall, these cells are pleomorphic, occurring in different shapes, that is spherical, ovoid, and filamentous. The mycoplasmas were studied for the first time by two French scientists, very important, so coming competitive exams. So this is a very important. First time by two French scientists, that Nocard and Roux, in the year 1898. Okay, the mycoplasmas were studied for the first time by the two French scientists, Nocard and Roux, in the year 1898. They were able to isolate pleomorphic bodies from the pleural fluids of cattle. So that cattle suffering from a disease called pleuropneumonia. Okay, so they were studied about the mycoplasma from the bodies of pleural fluids of the cattle. It's suffering from a disease that is called pleuropneumonia. So that the pleomorphic bodies were thin, spherical, filamentous, and stellate in the morphology. So the French scientists Nocard and Roux, they named the organism as Astrococcus mycoides. It's known as Mycoplasma mycoides. So such organisms were observed from several other sources also, animals, humans, and decaying organic matter, and were referred to as pleuropneumonia-like organisms for a long time. So that's why we are given that a shortcut name, that is a PPLO for the mycoplasma. Okay, PPLO means what? Pleuropneumonia-like organisms, PPLO. So for the long time that name was given by the French scientists. The genus name Mycoplasma for these organisms was coined by Novark. The genus name Mycoplasma this was coined by Novark and was later in the 1956. After the first, the Nocard and Roux in the year 1898, the French scientists, these two scientists were studied about the mycoplasma from the bodies of pleural fluid of cattle suffering from a disease of pleuropneumonia. That's why they given pleuropneumonia-like organisms, PPLO. Okay, so this is very, very important. Sometimes they were given as PPLO means what? Okay, so please remember this. And after that, the Novark and was later recognized in the preference to Astrococcus by Edward and Friend in the year 1956. So the genus name Astrococcus was discarded as it was the name of an algal genus. So that's why Edward et al. Proposed a new class, that is the mollicutes. The soft skin members, that is the mollicutes, for these organisms and separated them from the class Schizomycetes. So they were first given that Schizomycetes. After that, Edward et al. Proposed a new class mollicutes for these organisms, and after that separated from the class Schizomycetes. Okay, this is the introduction about the mycoplasma. So after we are coming to the characters of mycoplasmas, the mycoplasma showing the following characteristic features. It's a very, very important. See here in this, this is a diagram of a mycoplasma. These are common in animals. So cattle, sheep, goats, or dogs, rats, mice, okay, and also humans also. So they are associated with rheumatic arthritic diseases, infections of mammary glands and respiratory tract and urinogenital system. Okay, so we are observed on these all. So it's commonly, these are common in animals and also humans. These are associated with the rheumatic arthritic diseases, infections of mammary glands and respiratory tract and urinogenital system. So some mycoplasma members occur as saprobes in the soil, decaying organic matter, and in the human oral cavity. So some members we are observed in the saprobes in the soil, and decaying organic matter in the human oral cavity. Next, these mycoplasma-like organisms cause plant diseases such as sandal spike, astrellos, and mulberry dwarf, grassy shoot of sugarcane, corn, stunt, and potato, witch's broom. Okay, these are the very, very important. Some these diseases caused. Okay, some mycoplasma-like organisms, that is MLOs. Mycoplasma-like organisms. These are cause the plant diseases. Plant diseases are sandal spike, astrellos, mulberry dwarf. Please note down. Grassy shoot and shoot of sugarcane, corn stunt and potato, witch's broom. Okay, The cells are divided of cell wall and consists of only a plasma membrane. See here, this is the lipoprotein membrane is there, and soluble protein is there in the between, that ribosomes are present, only single DNA is there. Okay, soluble RNA is there. Okay, so the cells are devoid of cell walls and consists of only a plasma membrane. They vary in size from 300 nanometers about 0.2 micrometers in diameter. These are resistant to antibiotics such as penicillins and that act on cell walls but inhibited by tetracyclines and similar antibiotics that act on membrane pathways. Okay, so the cells are highly pleomorphic, form varying with culture conditions. Under different conditions, they appear as small coccoid bodies. Okay, that ring forms and fine filamentous, some of which are branched. So these are all characteristics of mycoplasma. Both DNA and RNA both are present. The mole present G plus C. This guanine and cytosine of DNA ranges from 23 to 40. Cells are usually non-motile. So because of flagella is absent, that's why these are non-motile. But some show gliding movement in liquid surfaces. So only in the liquid surfaces, some show the gliding movement. Okay. So they are gram-negative type, gram-negative. Most species require sterols and fatty acids in their growth media for growth. Okay, this is a mycoplasma. Most species form colonies that have a characteristic of fried egg appearance. So that mycoplasma form the colonies, that colonies have a characteristic of fried egg appearance. Okay, so in the classification, the mycoplasma classification, these mycoplasmas have now been removed from the class Schizomycetes. It elevated to the status of separate class, that is molecules. Okay, so it is in the now it is only that a single class, that is molecules. But before that, these under kept on the Schizomycetes class. But now this is separated from the Schizomycetes, and it is now this status of a separate class, that is molecules, meaning of soft skin. Okay, based principally on the absence of cell wall, the class molecules is divided into 3 orders. So mycoplasmatales and ecoplasmatales and aneroplasmatales. See here, these mycoplasma is the separate class molecules, and it has been divided into 3 orders: mycoplasmatales, ecoplasmatales, aneroplasmatales. Okay, these organisms require the cholesterol for their growth, which is incorporated into their cell membrane. The order includes 2 families: Mycoplasmataceae, Spiroplasmataceae. Okay, Mycoplasmataceae example is a Mycoplasma. Spiroplasmataceae example is a Spiroplasma. Okay, so these all are next. Ecoplasmatales do not require. So what is the difference between mycoplasmatales and ecoplasmatales? Ecoplasmatales do not having the require the cholesterol in their growth medium, but mycoplasmatales require the cholesterol for their growth. Okay, that is the difference. And aneroplasmatales are strict anaerobes occurring in bovine and ovine rumen. Sterol and phospholipids are required for the growth. That is the aneroplasmatales. Okay, so it's mycoplasma cause the serious diseases in human beings and animals and plants. Some human diseases are pathogenic mycoplasmas affect the respiratory organs and central nervous system, cardiovascular system, and urinogenital system in human beings. These are the human diseases. And some members of mycoplasma, pneumoniae causes the disease, primary atypical pneumonia. Okay, so these are called pneumonia-like organisms, CPLO. And some plant diseases also there. Some plant diseases are already discussed in that little leaf disease of brinjal, clover dwarf. Please remember this. In the exam, in the entrance exam, they will give these diseases. Diseases are very, very important. Little leaf disease of brinjal caused by, okay, Mycoplasma, Archaea, Actinomycetes. They will given as a multiple choice. So this is a little leaf brinjal, clover dwarf, clover phyllodes and cotton virescens, corn stunt, bunchit of papaya, witch's broom of legumes, and a stripe disease of sugarcane, spike disease of sandal, and aster yellow disease. These all are 10 diseases are belongs to plants. Okay, mycoplasma cause the 10 type of diseases in the plants. So animal diseases, mycoplasma mycoides causes the pleuropneumonia in the cattle, and mycoplasma causes the inflammation of genitals in animals, and agalactia disease of sheep and goat. That is these three are belongs to animals. Okay, so three are animal diseases, and 10 type of plant diseases, and some 5 or 6 types of human diseases: respiratory organs, central nervous system, cardiovascular system, and urinogenital system. Okay, so this is about the mycoplasma. Very important. These diseases are very important. And the next we are coming to the actinomycetes. Okay, actinomycetes. So it's very simple actinomycetes. Very smaller one. Actinomycetes are fungus-like bacteria. Fungus-like bacteria. These are called actinobacteria. Okay, actinomycetes is the fungus bacteria. These are called actinobacteria. Unicellular actinomycetes members are thread-like and resemble mycelium of the fungus. Okay, thread-like structures. See here, this is the diagram of actinomycetes. Thread-like structures resembles the true fungus. These are gram-positive bacteria. Okay, these are gram-positive. And reproduction is through fragmentation only. This is a vegetative method, that is the fragmentation. And based on the differences in the thallus structure, the actinomycetes members are divided into 3 groups. Okay, euactinomycetes and proactinomycetes and simple actinomycetes. So euactinomycetes, that a plant body is fungus-like branched mycelium and reproduces through the spores and conidia. These are called true actinomycetes. Okay, say it's a fungus-like branched mycelium and reproduces through the spores and conidia also. So same as it is fungus-like reproduction methods are there, that through the reproduces through the spores and conidia. These are called a true actinomycetes type. Okay, that is the euactinomycetes, U and true. Next, second one is the proactinomycetes. Mycelium development is nominal or limited. Okay, so in this type, that the mycelium development is normal or limited. They do not produce the spores. Reproduction is through fragmentation. That is the second one, proactinomycetes. And third one is the simple actinomycetes, unicellular forms with a distinct shape. That is the simple actinomycetes. Next, we are coming to the economic importance. Economic importance of actinomycetes. Many important antibiotics are synthesized from these actinomycetes members. So many important antibiotics are synthesized from the actinomycetes members. These are actinomycetes are aerobic gram-positive bacteria. Okay, so some antibiotics of examples of actinomycetes: streptomycin, neomycin, ritamycin. Okay, these are the antibiotics. Sometimes we hear that the type of antibiotics. Okay, streptomycin, neomycin, and ritamycin. These are the antibiotics. These are synthesized from these actinomycetes members. Many actinomycetes causes serious human and plant diseases, that is tuberculosis and leprosy. Okay, some diseases are coming from the actinomycetes, that is serious human and plant diseases, tuberculosis and leprosy. Okay, by the actinomycetes. So these whenever we are coming to the characteristics of actinomycetes, these are gram positive Some actinomycetes develop in mycelial stage, reproducing a unicellular specialized spores. See here, when we are observing of life cycle of actinomycetes, see here that substrate hyphae. So look like a fungus-like thread-like structures are there. Is aerial hyphae, and so coiling. So this is the coiling and separation, and these spore maturation. Spores are coming from these aerial hyphae. That is conidia or aerial hyphae. Okay, so conidia or spores are coming from these hyphae, and spore maturation, and after that spore released from that hyphae, pre-spore is there, round circle is there, and after that the pre-spore germination. Okay, so whenever the spores germinated, that substrate hyphae are formed. Okay, this is a vested growth life cycle of actinomycetes. Cell wall has some peptidoglycan as in gram-positive prokaryotes. A large number of other peptidoglycan types have been found in these organisms. So Berger's manual that divides actinomycetes into 7 sections, mainly that primarily based on properties such as cell wall type, conidia arrangement, and the presence or absence of a sporangium. Okay, that these are having the conidia, and that conadial arrangement and the cell wall type based on these characters that Berger's manual he divided into actinomycetes into 7 sections. Nocardiform actinomycetes and actinomycetes with multilocular sporangia, actinoplanets and streptomyces related genera. Okay, so 7 type of these are produced. Okay, so I will give you some economic importance of actinomycetes members. Streptomyces venezuelae, that species produce the chloramphenicol antibiotic. Chloramphenicol antibiotic, and Streptomyces aureofaciens chlorotetracycline. These all are important antibiotics. And Streptomyces garifalus cycloserine. Okay, this is giving the cycloserine antibiotic, and Streptomyces erythraeus erythromycin. Okay, venezuelae giving the chloramphenicol, and Streptomyces aureofaciens giving the chlorotetracycline, and garifalus species giving the cycloserine, and Erythraeus erythromycin, and Canamycetes. All are Streptomyces genera. The genus Streptomyces has nearly 340 species and 39 subspecies. So these are officially recognized Streptomyces or okay. They are best known for their synthesis of a vast array of antibiotics. Some are very important. Streptomyces griseus giving the streptomycin, and Streptomyces viridifaciens tetracycline. Okay, so and Streptomyces remosus oxytetracycline, and Naursi nistane, Streptomyces fradiae neomycin. Okay, these are all are very important of antibiotics of actinomycetes. The bacteria, mainly this is the aerial mycelium, divide a single plane from the chains 5 to 50 or more non-motile conidiospores in the chains at the hypal tips. The aerial hyphae possess the extra wall layer or sheath. So the substrate mycelium when present does not undergo fragmentation. The spores may be smooth, hairy or spiny in texture. The aerial mycelium is colored. Okay, so this is the life cycle of actinomycetes. These are very important bacteria, actinomycetes and mycoplasma. The mycoplasma that diseases are important, and actinomycetes that antibiotics. Okay, so that some species are giving the antibiotics. Some area that questions coming area is that antibiotics area. And in the archaebacteria, that type of archaebacteria, alkalophils, acidophils, halophils, thermophils. Okay, that is in the archaebacteria area. Okay.
Madam, thank you very much.
Okay, sir.
Okay. Tomorrow
Thank you.
evening, tomorrow also we will plan to evening, madam. Okay.
Sir, hello.